The brain is made of billions of neurons and controls all your complex behaviour. 3 parts to know: the cerebral cortex, the outer folded layer, responsible for consciousness, memory, and language; the cerebellum at the back, which controls balance and coordinated movement; and the medulla, which controls unconscious actions like your heartbeat and breathing. Scientists study the brain by looking at patients with brain damage, also by stimulating areas electrically and by using MRI scans. In the exam, match each region to its job: the cortex for thinking, the cerebellum for coordination, and medulla for the automatic stuff. The eye. The eye is a receptor for light. Light enters through the cornea, which does most of the focusing. It then passes through the pupil, whose size is controlled by the iris. The pupil is focused by the lens onto the retina at the back, where the light receptors send signals down the optic nerve. Accommodation is how the eye changes the lens shape to focus on near or far objects. For a near object, the ciliary muscles contract and the lens becomes fat and rounded. For a distant object, they relax and the lens becomes thin. In the exam, there are 2 defects to know. The first is being short-sighted, where distant objects look blurred and is corrected with a concave lens, and being far-sighted or long-sighted, where near objects look blurred, and this is corrected with a convex lens. So near needs a fat lens, far needs a thin lens. Controlling body temperature. Your body temperature is monitored by the thermoregulatory centre in the brain, which has receptors sensitive to the blood temperature. If you're too hot, you sweat, which cools you as it evaporates, and the blood vessels near the skin widen. This is called vasodilation and is needed to lose more heat. If you're too cold, you shiver, which releases heat from respiration, and the blood vessels narrow. This is called vasoconstriction, to keep heat in. Water and nitrogen balance and the kidney. Separate biology covers how you control water. Your kidneys filter the blood, removing urea, excess water and ions, and making urine. Urea comes from the breakdown of excess amino acids in the liver, a process called deamination. Water level is controlled by a hormone called ADH, by negative feedback. If you're dehydrated, more ADH is released. The kidneys reabsorb more water and you produce a small amount of concentrated urine. People with kidney failure can be treated by dialysis, a machine that filters the blood, or by a kidney transplant. In the exam, remember ADH increases the water reabsorbed. More ADH means less urine, and that urine is more concentrated. ADH equals add water back, more ADH, more water saved. Separate biology adds 2 more hormones. Adrenaline is released by your adrenal glands when you're frightened or stressed. It increases your heart rate and boosts the delivery of oxygen and glucose to your brain and muscles, ready for fight or flight. Thyroxine from the thyroid gland controls your metabolic rate, which is how fast the reactions in your body go. Thyroxine is kept steady by negative feedback. In the exam, remember that adrenaline is the fast, short-lived fight or flight hormone, while thyroxine sets your background metabolic rate and is controlled by negative feedback. Plant hormones. Plants respond to light and gravity using a hormone called auxin. Auxin controls growth in the shoots and roots. In a shoot, Auxin gathers on the shaded side, and it makes those cells grow longer. And so therefore the shoot bends towards the light, which is phototropism. Shoots grow towards the light and against gravity. Roots, however, grow towards gravity. We use plant hormones in farming, as weed killers, as rooting powders for cuttings, and to control fruit ripening. In the exam, a required practical looks at the effect of light or gravity on seedlings. Set up a fair test and change only the direction of the light. Auxin builds up in the shade, so the plant bends towards the light. Protein synthesis. Separate biology explains how a gene actually makes a protein. The DNA stays in the nucleus, so a copy of the gene is made, called messenger RNA. And this is able to carry the code out to a ribosome. At the ribosome, the code is read in threes, and each triplet brings a specific amino acid. The amino acids join up in order to make a protein, which folds into its final shape. A mutation can change the code and so change the protein, which may stop it working. In the exam, the two stages are transcription, copying the gene into messenger RNA, and then translation, reading that code at the ribosome to build the protein. So transcribe means copy, and translate means to build. Copy in the nucleus and build at the ribosome. Evolution, speciation, and the history of an idea. Separate biology adds the story and the detail of evolution. Charles Darwin proposed natural selection after his voyage on the Beagle, and Alfred Russel Wallace reached a similar idea independently. An earlier scientist, Lamarck, was wrong. He thought characteristics gained in a lifetime were inherited. Speciation is how a brand new species forms when two populations become isolated, face different conditions, and change so much that they can no longer interbreed. In the exam, the test for a separate species is that they can no longer breed to produce fertile offspring. Trophic levels and biomass. Separate biology measures energy and biomass in ecosystems. Each stage of a food chain is a trophic level. Level 1 is the producers, which are plants, and they make biomass by photosynthesis. Then come the primary, secondary, and tertiary consumers, and finally, apex predators at the top. Decomposers break down dead material at every level. Only about 10% of the biomass passes from one level to the next because energy is lost as heat from respiration, in movement, and in waste like urine and feces. In the exam, you can be asked to calculate the efficiency of transfer. It's the biomass passed on divided by the biomass taken in times 100. Roughly 10% moves up each level. This is very important to remember, and the rest is lost on the way. Food security and farming. Finally, feeding a growing population, which is also known as food security. Biomass transfer is so inefficient that we can produce more food by being efficient with it. In intensive farming, animals are kept warm and their movement is limited, so less energy is wasted and more goes into growth. Biotechnology helps too. For example, growing mycoprotein from a fungus to make meat substitutes, and also genetically modified crops. These methods raise yields, but also raise concerns about animal welfare and the environment. In the exam, limiting an animal's movement and keeping it warm both reduce energy loss, so there's more biomass which goes into growth.